appreciate the invitation. It's a pleasure to learn about all the other interesting work going on with these devices. I'm going to talk about integrating near field and electron optics, what I call kind of dual vision. And as I mentioned, I am at NREL, but this work was basically all done at the Naval Postgraduate School in Monterey. So it is not the Naval Research Laboratory. It is not the Naval Academy. It's in ourselves referenced. It's in a much better location. It's in Monterey. It's the Navy's graduate school, but it now serves all areas of the armed forces, as well as colleagues from about 35 to 50 countries at any given time. It's kind of an interesting place if you ever get a chance to go there. About 2,800 students, 1,400 of them in residence, 185 million in reimbursable funding at last glance. And there's the NREL disclaimer. This is where the work was done. I have then moved there. Okay? I moved the system with me. And many of you are probably familiar with NREL, and we are certainly hoping to get this activity going there as well. So I'm going to claim that I'm giving one of the first actual dual beam talks, right? And we're going to use the electron beam as one specialized, I'll admit, but still a multi-probe. So I'm going to talk about what you can do with this, tell you a little bit about what I have found in the literature on the integration of SEM and NSOM. And then my focus is what I'm going to call transport imaging. That is, what I really want to look at is the transport of energy, whether that's as carriers, in my main interest is semiconductors, materials and devices, or photons. Some related examples in other areas, and then kind of looking ahead, what might we see. So by the end, I want you to understand pictures like this, which maybe are a little different than what you might initially think they are. Now, as you well know, if you've done these things, even with one probe, you've got a whole range of options, right? You can bring your illumination in from the top and look below. You can bring the illumination from above and collect. You can, if you've got enough signal to get away with it, try to both illuminate and collect through the same tip. Good luck. Can be done, but you better have some good signals. You can illuminate in the near field and collect in the far field. You can illuminate in the far field and collect in the near field. You can do an amazing range of things, and I'll show you some different examples. But in all cases, when you basically have a single probe, you're going to assume to first order that you're collecting from where the light is generated. And that's an assumption. It may be a very good assumption, but it is an assumption nonetheless. And I want to work at the point where that assumption sort of breaks down a bit. So many of you, I'm sure, if you've been in the semiconductor field, have seen images like this. This is a CL image, or scanning PL would look the very same way. And people look at that and they say, well, that's spatially resolved luminescence. True enough, okay? But what you're doing in an image like this is you're scanning the electron beam or the laser back and forth, and you're looking at the light that's emitted when the beam is at any particular spot, okay? And then you're going to assume that the light basically comes from exciting at that spot. Well, that's true, but not exactly. And the fact that it's not exactly true is shown very clearly here. This is actually a spatially resolved luminescence plot of the light that you see, and I'll talk about the material later. It's very high quality 3.5 material. But basically when the electron beam is at a point right in the center of that distribution. So that actually is where the light is coming from. And notice this is 100 by 100 microns. Okay? So what you're seeing there is, of course, what you know happens in these materials, and that's carrier diffusion, which in these materials can be quite extensive. So although you're generating at a very tiny point, and remember, I'm going to use an electron beam, so my generation volume can be 100 nanometers or less. The actual location of the luminescence is quite broad. Okay? So that's what I mean by transport imaging. I want to image the transport of charge, and I don't care whether it's carriers or photons or whatever, from their point of excitation. Okay? So that's what we're going to try to do. And that gives us additional information. These pictures are still interesting. They tell you something about the spatial uniformity of your material. That's important. But they don't give you quantitative information about local transport properties. Okay. Oh yeah, so imagine then my electron beam. It's a point in this particular image. In this particular image, what we've done is we put a field on, we've drifted the charge. In this case, my point is going to be right there. So I need you to stop to think about scanning electron microscopes as always scanning, all right? Sometimes it's going to scan and we'll take a nice picture, but sometimes we're going to use it as a point source or as a line source. So we've been doing this in the far field for a while. And again, here's an image, and this is not a simulation. This is actually spatially resolved luminescence. And we did this initially just by putting a CCD camera and an optical microscope into a very old but very functional JEOL 840 SEM. And I won't go through all the details because I want to focus on the near field work. But basically, you collect pictures like this, which tell you a lot about the mobility lifetime product of your material and carrier transport. And then you can fit the data. So here's the data for 3 different materials on log scale, function of distance. Over here is the black are the data points. Okay? The solid lines are the Bessel function fits for diffusion from a point source. And as you can see, you can get really high quality fits and therefore extract with one image minority carrier diffusion lengths, which are very important for people that make a lot of devices and of course in particular solar cells. Now you can do this in all kinds of geometries. You can use a point source on a nanowire, and it's quite nice, and it's an exponential decay. You can do a point source on a thin film. If you're smart after a while and your material is uniform, you realize you should be doing a line source and integrating. You can do point sources on bulk or line sources. This is in the assumption that the surface recombination is small. So we don't have to have a heterostructure, although a lot of times if you do, you can do some very nice work. But you don't have to. If S is not small, if there's significant surface recombination, then you can produce a chart like this, but some of the equations get too big to put on the picture. Okay? They're more complicated, but they can all be derived and are in various places. And you can extract the transport parameter you care about. So some of the things we've done in the far field, this is kind of one of the classic examples. This is an image of anisotropy in an ordered alloy. So these are ordered alloys, gallium phosphide. If they're ordered to enough extent, then the effective mass is anisotropic. And hopefully you can all see that that's not a sphere or a circle, right? It's an egg. And the egg reflects directly the anisotropy of the transport. We can fit this diffusion length in all directions, extract these. And so again, with a single picture, we've extracted minority carrier transport in its anisotropy. More recently, we've done work like this again in solar alloys of interest to plot minority carrier mobility. That's a hard parameter, right? Majority carrier mobility is easy. You do a Hall effect measurement. But the minority of the mobility of minority carriers is much tougher. And the people who get at it usually get at it by going all the way through device modeling. We don't have to do that. Again, we take a picture, and then we work with our TRPL colleagues to get the independent lifetime. You put that together, you have a direct measure of the mobility. Just focus here, minority mobility. This is in gallium phosphide again. It's the top material in a triple junction solar shell as a function of temperature. Just for comparison, the blue and the black data are majority mobilities. Okay? The minority carrier mobility has a very different temperature dependence. This has been seen in gallium arsenide and silicon. These are the first measurements in gallium phosphide. So anybody who's telling you that you should use majority carrier mobility as kind of a good enough reference for minority may or may not be correct, right, depending on the temperature of interest. So we do things like that, and that's what I mean again by imaging transport. In this particular case, we've taken a planar structure, put some contacts on it, put an AC voltage going back and forth, and again, we're imaging the drift of minority carriers, and I can't actually remember anymore if this is P-type or N-type, but whichever way it is, these are the minority carriers drifting back and forth. Again, high quality material, 400 microns. Now, I can either wait, you can ask, or I can tell you the little reflect, so the beam is right in the middle of that red spot, and it's at a point. Okay? And then we have a little reflection, and you saw there was a defect there where the quality goes down. We can see all of those things. And again, if you want to stop me along the way, feel free. So we want to be clear about what the pictures are. We're not doing scanning. But now we're going to move to the nanoscale. So suppose we want to do this now, not on a thin film or a bulk sample. Suppose we want to do it on a nanowire or a nano belt, or the contrast here isn't very good, but this is a triple junction solar cell in cross section. If we want to do that, then my far field camera is not going to do the job. My resolution on that is about 350 to 400 nanometers per pixel, depending. So we might expect minority carrier diffusion lengths in structures like these of only a couple hundred nanometers at most, and therefore we're going to need higher resolution collection. So for that purpose then, what I want to do is integrate an AFM ensom into the SEM. Why? Well, I need a couple of things. One is I need to see my structures, and it's a heck of a lot easier to see it with an SEM. There's nothing better. I mean, the scalability of electron optics is just incredible for this purpose. We can look at a nanowire one day and we can do a full wafer scan the next, right? SEMs are great about that. And then I need an ensom to collect my light. We can do this in materials with any luminescent signature. But I need the option to scan the collecting probe. Scanning the sample doesn't help me if I want my beam in a point source on my nanowire. If my nanowire is doing this, that's not the experiment I really want to think about. So I need the option for probe or tip scanning. Sample scanning is nice too. We make CL maps as well, but for the real transport image, we need it. And therefore, when I started to try to do this in about 2006 or 7 or whatever it was, that led me to the Nanonics system and a phone call to Israel that probably got Aaron up at some ungodly hour to talk to me about whether we could do this with his system. Now we can. So this is, I've shown you pictures like this in the far field. This is one now with the ensom. So the electron beam is coming down and fixed at a point. And again, I can make that of various dimensions. In the center of that, and then this is the light. This is the ensom image of the diffusion of carriers within that system. And of course, my little dip here, which I don't quite know how, is the probe tip, right? Remember, I've got a probe tip now scanning across this image, however you want to think about it. If you want to think about it this way. So it's going to block, it's going to block, it's going to block. When I get on this side, it no longer blocks. It simply maps the recombination of the source from a point. So this is, again, electrons, or holes, I can't remember, whichever the opposite one is. These are p-type materials usually, so we're usually mapping electron mobilities. We can also do the same with photons. We can generate light. These are, I'll talk about this work a little bit later in more detail, but these are actually they're pretty large, but sort of nano rods, if you want to think of them. The electron beam is sitting somewhere down here at a point, and these are the photons that are emerging out the other end. So whether your transport is via electrons, holes, photons, or, as you've seen a lot of the nice stuff today, plasmons. Now, a number of people have put dual probe SPMs of various types into SEMs. And there's some pictures from a number of references in the early 2000s. But their main goal for the SEM was to be able to see, right, their dual probes, where they were and how close together they were and where they were on their structures. And that's, of course, a very handy thing to do, and you can do that in my system as well. But that's pretty much the primary extent of what they wanted from the electron beam. So I want a little bit more. I want to use the electron beam to generate my electron hole pairs, for example, if I'm doing more traditional semiconductor work. I need an AFM to maintain my system in the near field, and then, using the probes that we're all familiar with, I'm going to do ensom to collect them. So you can do this in a lot of different ways, and actually in the very earliest papers that I found, there are a couple groups, I'll show you the references here in a minute, that did this. What they were interested in was near field CL. They wanted to do CL mapping, just like the things I showed you earlier, only they wanted higher resolution than you get with traditional CL. And so they designed their system to scan the sample. And we'll talk about that a little bit more, some of the pros and cons and how far you can really push the resolution, but that was with sample scanning. The other thing you can imagine is scanning the E-beam, and there was some work done on that. You can do some imaging of diffusion of carriers. You can image the beam interaction in an SEM. You can do the near field CL. But the one I really want to focus on is this, where we're going to fix the electron beam and then scan the NSOM probe. And that's where you can do a range of things that you can't do with that other approach. Now, in this very nice Review of Scientific Instruments article on this dual probe scanning NSOM, this is a very nice picture, I think, that kind of summarizes, right, what I've just been talking about, that you're going to have diffusion of these things that may take excitation from one area and lead to light collection in another. And depending on what you want to do, that's either a limiting factor or something you can take advantage of to look at physics of interest. So these are two of the earliest papers for integrated AFM NSOM, and they did do some NSOM as well, some optical collection. But again, sample scanning by moving the sample and keeping the collection probe near the E-beam. So some nice work, but pretty limited. And actually, if you look at the downstream history of it, it hasn't really had a huge impact, and I think we'll be able to see why in a minute. The original driver was to get better resolution from your cathodoluminescence. Everybody who does CL knows that an electron beam coming in creates a generation volume. The size of that volume depends strongly on the excitation energy. So here's 20 keV in silicon. You can see you actually, no matter how tiny that little guy is, you excite secondary electrons and everything that follows, in this case over a range of a few microns. Now the trend since this early work is that people are now doing 5, 3, even 1 keV. And with all the improved optics, that generation volume can go very small, but you lose signal too. Okay? So in traditional CL, you bring the beam down, you generate light from that area, you collect it all, you throw it to the detector, and you say, that's my signal. And although your beam hit there, your signal actually grows. Now if you look at the problem more carefully, there's multiple relevant length scales that determine your resolution. Because in the early papers, they'll say, well, the reason you want to do near field CL is that you can limit your collection volume to the probe tip, right? So the generation volume is 1, right? The sample is a big deal too. It makes a big difference whether you've got a bulk sample or simply a quantum dot, for example. The carrier diffusion length, and then the optical absorption coefficient for luminescence. You generate light here, right? But depending on your sample, that light may propagate out, be reabsorbed and re-emitted. So when people ask, well, what's the resolution you can get with near field collection in CL? Again, like most important questions, the answer is it depends a lot. Okay? If you've got an extended source and a generation volume, and you simply do standard CL, then yes, it is the generation volume. But if you have a very localized source, then you don't care that much what the generation volume is. You care about the size of your sample. Okay? So if you could imagine having a single quantum dot that luminesces at a different wavelength, you may well be getting information just from that. But now I'm going to ask you to add in this idea of energy transport, which could be either by diffusion of carriers or by photon emission and reabsorption. And in that case, you can have an even larger volume than the generation volume, or smaller. Again, all of this is going to depend on your particular sample. And if you have the collection that a lot of people care about, which is a collection of nanostructures, then your honest resolution is some convolution of the whole mess. And that doesn't change that much. It changes to some extent, but not completely, if you bring in a collecting tip of small aperture. Okay? So yes, it's going to help you, right? But on the other hand, if you've got energy transport so that you've got some generation over here, and the photons for that can get back to here and go in the tip, then it's not as simple, right, as to say that it's the size of the tip. Now, you're counting on the fact that if we go back to my picture, and I won't take you all the way back, but again, remember that distribution. It's fairly steep, and so it's a good assumption, but the combination comes together. So with that in mind, my focus isn't so much higher resolution CL. They're doing incredible high resolution CL now in STEM systems. If you're willing to prepare your sample like that, I think that's the way to go. I'm interested more in transport imaging. So I got a new SEM, tremendously. Okay? Here's the Nanonics system. The fiber comes in through a feed through here. The detectors are sitting up here. Here it is, getting ready to go inside. There's the pole piece, and the electron beam is going to come down onto the sample and be my generation source. And so you could say, and many people do, well, geez, just do this with a laser, right? Who needs them? And you can, but there, again, I want to argue that there are some nice benefits. The one is a very tiny generation volume if you do it right. The other is the overall scalability of the system. And the third is, if we're interested in semiconductors, we can do small band gaps one day, diamond the next. We don't really care how big the band gap is. So we have some nice capability there as well. Okay. Let me show you a little video of how this works. Just be a little bit patient, but I think it'll run. So what we're seeing here, okay, so there's my NSOM tip. This is a sample with some nanowires. We've got the grid to help us do some references. I'm finally now at a magnification where my nanowire appears. We can keep zooming in. Tip's been in a little while, you can see. Right? It didn't look like this when I started, Aaron, I promise. You never think to take the pictures first. You don't take the pictures until you're done. My nanowire, again, my SEM is obviously still scanning, but when I start to do my experiment, that will stop, and I will stick the beam at whatever point I choose, but just for illustration purposes. So this is how then I will take my data. And I can't take data right where the beam hits, but I can take it darn close next to it, and I do some filtering. There are some things that we have learned painfully over the time, like if an electron beam hits the tip, it generates light for sure, okay? So you have to take that into account and do some filtering and things like that. All right. So what should you see if you do that? Well, there's a couple examples. Again, the beam's going to come down. It's going to generate a lot of luminescence right there, no argument. But some of those carriers are going to diffuse in both directions. Maybe not far, but depends on the material. They're going to generate light. And the other thing you're going to see is that these photons get beautifully wave-guided. These are waveguides. So you would expect to see some light coming out the other ends as well. Okay? And in fact, that's exactly. So here's one image. The electron beam is at my famous green point right off the edge of the sample. So I'm off where I start to collect, and then my probe tip is going to scan here. You notice what we believe is carrier diffusion due to recombination along the way. We get a little backbone luminescence that I think is leakage out of the thing, and then you come down to the end, and there's your nanowire as waveguide. So photon transport, electron or minority carrier transport, etc.
Are you seeing oscillations along the backbone there? Is that real or artifact?
I think it's artifact, although I would like to pursue that with higher resolution and more experience now.
How do you differentiate majority versus minority carriers?
So usually if I have dope structures, then they're minority carriers. You're correct that if it's a very low doping, or if I go into a high injection regime, then it would be ambipolar transport. We work very hard, in the earliest work I showed you, we work very hard to make sure we are in a low excitation regime, meaning that, you know, if it's, let's say the material's 10 to the 17 N-type per cubic centimeter, we only want to generate 10 to the 14 or 10 to the 15 per cubic centimeter carriers. Then it's all minority. But we've worked a lot in some insulating materials, and then it's a much more difficult problem. All right, so those are the pictures that we kind of see. And here's another example. So again, the electron beam is up here. This is the NSOM image Again, basically similar to what you saw before. There's the wave guiding, there's what we believe to be the carrier diffusion. Our AFM images are not great. They've gotten better over time than this one, but you know, we're convoluting these two things, so we're not trying to get high-resolution AFM, although we should do better. But that at least gives us the axis for which to compare, and then we can make these pictures. Now, my interest, though, is not just nice pictures, fun though they are, is to get some quantitative information about transport, particularly in my new life as a solar cell person. But here's an example of that. So we worked in collaboration with colleagues at Sandia. These are gallium nitride nanowires. And then they put different, not really coatings, I think of them more as core shell structures. So InGaN on the outside, AlGaN, and then nothing. So indium gallium nitride has a smaller band gap. Aluminum gallium nitride has a bigger band gap. It should be more like a heterostructure, traditional heterostructure, and then the bare wavelength. Okay? If you do standard CL, these are the spectra that you see, and they're on log scales, just so you get a sense. But you know, here's the near edge peak, and you notice, again, not too surprisingly, that when you have a nicer heterostructure, you get a pretty decent near edge peak. Still visible for an uncoated one, but much weaker. And then with the InGaN there, almost like a surface sink, right? And so very little. And we're used to doing things like this, but from a transport point of view, it still leaves you saying, so what, right? Give me something a little more quantitative for transport. Well, now we can go in, take pictures like this, and plot the intensity as a function of distance from the excitation point. Put it on a log scale, exponential, if this is a good quasi-1D system. And so if we compare similar diameter structures, we get, again, these minority carrier diffusion. These are all doped wires, about a micron, 660 nanometers, 350. So a way to try to get inside the transport. And of course the question is always, well, is it bulk, is it surface? Well, it is what it is, because that's how far the carriers are going to go. So until people start making things without surfaces, the surface is part of the picture. We have been able to do this only in the far field work, but...
I'm sorry, I have a question about that. You mean, you're collecting, the height of that structure is about what, about a micron, or is it,
these wires are about half a micron.
Half a micron. So basically, most of the light that you're looking at is coming from the very surface. Meaning, I think. I don't know
yeah,
how
although it's a
deep-
little bit below band gap, so I would say it probably propagates more than that.
Okay.
It's below, when we get the spectroscopy, it's below band gap enough, but I think it's probably more. Actually, in standard CL, you would assume your light is coming from a micron or so down.
Yes, but what happens is that a near-field optical probe has a very large angle of so that the amount of light that's collected is biased to near the probe. So you're seeing mostly, I would say, 100 nanometers, depending on the size of the probe, 100 and 200 nanometers.
These are about 200 nanometer
Okay,
diameter.
maybe
And remember, these photons are coming
Deeper.
straight up. They're not the ones
it
propagating.
could be, right.
Yeah. These are the ones that are recombining along the way.
Okay.
Okay? So it's like having multiple generation sources all down the
Right,
wire.
right, that's true.
I mean, you're correct that I've got this other light that's working its way, but hopefully
Yeah,
that
of
is,
course. But that's not producing more electrons.
no,
That's just guiding photons.
that's right. Yeah, absolutely. So we have not done this, well, actually I have an exception at the very end. We are just starting to do this, but in the far field, if you put some bias on these things, you can see drift as well. And so this is where the dual probe would be interesting. We would be able to not only, dual probe plus, right, and we'd want at least 3 or 4. You'd be able to go in, generate, collect, and electronically excite your structure with bias or whatever you want it to do. So this is, again, earlier work in the far field. Generation point right there, you see the diffusion in the heterostructure, and then as we bias, you see the drift, and you can fit the structures, the math is very well known. You can fit this and extract diffusion lengths. And if you put an AC field on, you can watch it swash around back and forth. You can take nice pictures that way. And the goal of this work was actually to go in and get minority electron mobility in very heavily p-type materials, because people were interested in some of this for some of the transistor work. So there was some theory work around, but no real measurements. We were able to make the first measurement. That's the theory. And again, just to remind you, that's the majority electron mobility. It's very different. So the numbers are important for some devices. Different photonic example. I showed you these pictures before. What we've done here is take, these are zinc oxide nanowires, I believe. So zinc oxide, and then, again, we have an SEM, so we can go put the beam at any point that we want along the way, generate carriers, and then look at the intensity of the light that's emitted from the end of the wire. So what this allows us to do, actually, is a Beer's law experiment, where by varying the location of the excitation point, we can measure the absorption coefficient directly in an individual nanowire, which again it could be done, I think, but it's challenging to couple light. And so here you can go in and generate the light wherever you want. We did that. We decided to look at some different diameters and got an interesting result that the coefficient that we measured, the absorption coefficient, the alpha, actually varied with the diameter of the wire. Okay? And when we plotted that, we saw, so there's I'm calling it the attenuation coefficient, because it's not just absorption in the traditional sense. Attenuation is a function of the diameter of the wire. The bigger the wire, the lower the attenuation coefficient. Okay? Slope around d to the -1. One very simple explanation is that the light that is being wave guided down the structure encounters the surface more as the diameter is smaller. So if you kind of model what the number of bounces is per unit length, it goes like 1/d. And that's probably due to the fact that the surfaces aren't perfectly ideal. So we can get the number, but we see a dependence there through this technique. There's an interesting paper. They were looking at, and again, you see this kind of phenomenon when people do more standard PL imaging. But they were making nanowire lasers, and this is the nanowire length in microns, the nanowire diameter, okay? And so if you just focus on the diameter, black solids indicate that they got the thing to lase, all right? And the open circles indicated that it didn't lase. And you notice, obviously, that the thicker ones were able to lase, the thinner ones did not. This would be one explanation, that effectively your internal alpha may well go up with diameter. So it may explain that information. Let me just spend 5 more minutes or so on talking about solar cells. If you're from NREL, you have to show this picture. Many of you have probably seen it. It is the famous NREL maintained plot, but from labs all over the world, of solar cell efficiencies as a function of time. The highest records are held, of course, by multi-junction cells, where they grow. In this case, gallium indium phosphide, gallium arsenide. The lower cell historically has been germanium. These now have efficiencies of 44 to 45%. They're used primarily on satellites. They're also of interest for concentrators. So you take all that sunlight and put it into one very small but very expensive cell. Here's the perovskites, if you're paying any attention to that story. Perovskites are making a lot of these people very nervous. So we're watching that. That's a very interesting story. But anyway, let's talk for a few minutes about triple junction solar cells. The idea, of course, is you're going to grab different part of the spectrum. And so one of the things I want to do is think about doing transport imaging now in a structure like this in cross-section, to really watch the motion of energy within this. They're 45% efficient now. I think the theoretical limit is about 60-something, depending on the structure. But in the solar cell business, every percent matters. So if you could capture where some of that other energy goes when you lose it, that would be good. So what we've been doing most recently is trying to, again, start to do this in cross-section. So we're going to take a solar cell, cleave it, and then look in cross-section. Here's the device, and you can see it here in the Multiview 2000 getting ready to go into the SEM. And if you look carefully, you'll notice it's red. Right? That's because we actually do have leads on it, and we're forward biasing it. We forward bias a solar cell, it's an LED. So this is the way we sort of, very expensive LED, but an LED nonetheless. So this is the way we check to make sure we have electrical contact before we go in, and then we're all hooked up. So it's going to go in. We're then going to bring the electron beam down at a point, okay? And I can stick that point here. I can stick it here. It's not going to be terribly interesting if I put it there because germanium doesn't have much luminescent signature, at least not enough for me to pick up yet, but in these. Now before we do that, we do all the things you ought to do. We do spectroscopy, we do CL mapping. And this is this is a PL map. And this is data I actually took during my Fulbright stay in Israel. This is an ensemble luminescence PL map where I'm going to do what I told you I never do, which is scan the sample. Okay? So this is a standard map. You see the indium gallium phosphide. This is the region of the junctions, which do not luminesce. There's a lot of carrier recombination. There's a center cell. The third cell would be to the back. So the first thing we do is make sure we understand the light that we're looking at. And this kind of gives you a sense of some of the resolution. I'll show you the comparison in a minute. So there's the intensity as a function of position. Again, this is with a laser beam coming down, scanning the sample, and collecting all the light. It's not the experiment I want to do, but it's a step along the way. I know there's going to be some discussion tomorrow about near field excitation, far field collection, or vice versa. I just thought I would show you this for comparison. This is the same structure, okay, where in the one case, I'm doing near field excitation and far field collection. In the other case, far field excitation with a laser beam and collecting, all right, with the near field. And you can see the comparison. We do get better resolution with the near field excitation. And the reason for that, I believe, is this issue I showed you earlier of the charge carrier transport. That if I generate here, we know that light is emitted over here. We've seen that other ways. So this is the better one. I get a good sense of where I'm going to put my structure. And now I'm going to turn around and do actual transport imaging. So here's the structure in cross-section. The contrast is a little better than I can see here, but the gallium indium phosphide layer is here, the gallium arsenide layer is here, and my electron beam is going to go to either of those positions, and then the tip is going to come up and look at the luminescence from excitation at those points. And again, I'm going to use some filters so I know which wavelengths I'm looking at. This work is very recent, and I'm still talking to my collaborators, so I can't show you too much of it, but just a little bit of a teaser here. So this is the same data in 2D and in 3D. Again, the electron beam is here, okay, at a point. And when I scan, you see that I get luminescence, again, due to carrier transport, through this entire gallium arsenide layer, bounded by the tunnel junctions. And that means, of course, that the solar cell is doing exactly what a solar cell should do. These are high-quality materials. Carriers are diffusing throughout that whole area, making it to the junction, and some fraction of them, however, are recombining along the way. Okay. So frankly, when I see photons, but I'm doing it in a rather unique structure, right? I cleave the sample and I'm looking for them. But solar cell people don't want to see this. They want carriers, not photons. But we're going to be able to analyze this data in high resolution. We can see some recombination areas in their interfaces and things. So this, I believe, is the first real transport image in cross-section on a solar cell. If you put the beam, WSXM kind of flips everything, right? Those of you who have used it. So it's different than my physical layout. If I put the electron beam over here in the gallium indium phosphide, that's the wide band gap layer. I get luminescence, but what I'm mapping here is the gallium arsenide photoluminescence, 880 nanometers. This is light generated in the gallium indium phosphide, which is red, and then the fraction of it that goes this way gets reabsorbed, right, by the lower band gap material, re-emitted, and produces 880 nanometer light. So this is what is called photon coupling or photon recycling in the solar cell world. And the state of the art of this stuff now is such that they are actually figuring out how to minimize and recapture this kind of emission at high efficiency. Because again, even a few tenths of a point in this business is important. So we can actually look in literally now and see it, see the beam here. So imagine the photon here, a stream of photons here, and then light being emitted in the neighboring stuff. So going forward, where do I see this? Well, I think there's very limited application for near-field CL that drove the original papers. Why? Because if you watch what people are doing with STEM now, so these are CL images associated with, I think these are photon or plasmonic kind of things with silver nanoparticles. But this is 200 nanometers, okay? These are wavelengths, so forget those for a minute. Look at the scale bar of 200 nanometers, and there's the spatially resolved luminescence as they scan the beam. I mean, that's pretty impressive, and it's gotten even better since that point, and we're still limited by the size of our probe. The smallest ones I use are about 150 nanometers. I've made it to 100 once or twice, but I usually have to use larger ones. So,
so like, how big are these two? Oh, this is the structure is
there it
like
is.
200. So it's a highly limited
Yes,
structure.
absolutely.
Yeah.
Absolutely. But there's just a lot of work like this now. It's growing, and of course you need an STEM, but they're out there. And I just think we're not going to, I wouldn't be interested in trying to compete with that. I want to do what we do differently. So, but their resolution, our resolution are going to be limited by transport and by throughput. I think this is interesting stuff to watch. However, I do think looking at transport of other kinds of excitations can be very interesting. This is some very nice work. Pullman's group is doing a lot of CL plasmonics work, but they're scanning the beam, okay, and they're seeing the maps. And you've seen some nice maps earlier, PL maps and others. The electron beam can generate plasmons, and although I have not done any of this yet myself, I think, imagine now going through and mapping, able to look at the full excitation when the excitation point is moved all over.
Excuse me. In the plasmonic one that you just showed, this paper, this is nanometer with SEM.
Right. This is cathodoluminescence. It's E-beam generated plasma
Plasmons.
mapping.
Yeah.
That's right, with the beam scanning now.
Scanning.
Right. Yeah, absolutely. And polarization resolves CL detection.
So in principle, you could, in fact, even close to a plasmonic structure, have an electron beam, because it has an E field, which could in fact generate plasma.
Yeah. Right. So we should be doing this. We need the right
So
structure.
this could
But
be
yeah.
very interesting.
But it shows here kind of what they're taking advantage of the generation. But the assumption is always, and again, it's a good assumption. I'm not arguing that it's not a good assumption. I'm just saying there are limitations to it. Some of you may be familiar with Pullman's work on angle resolved CL. It's really gorgeous, and he uses it to show how the emission from different materials differs in its angular resolve. So what he does is he uses a particular kind of mirror and imaging so that the light that comes out at different angles can be imaged, okay? But again, note the assumption. The assumption is that the light all comes from the point of generation. To the extent that this is not true, then it muddies the water a little bit for this kind of work. So these folks need lousy materials where the charge doesn't go anywhere, right? In many cases that might be true, but in other cases it would not. So my argument simply would mean that dual probes, whether it's an E-beam and an NSOM probe, or whether it's going to be an E-beam, or whether it's two NSOM probes, whatever you want it to be, you can really test the assumptions of localized emission and study all of the related physics. And I think if you could apply local fields and then watch and manipulate these things, you can do even a lot more. And this is one of the papers where that's done with two standard ensom probes, no E-beam here, but illumination with one collection with the other, generating and imaging the surface plasma polaritons. So this is being done. The question is whether with the E-beam you kind of can see all the structures and move around with ease. Now, what are you going to do if you don't integrate it yourself? Well, every time you talk about this, people will tell you, oh, I know companies that put AFMs in. They do, but this is one example. You can scan the sample. You can't scan the probe. And again, that would limit many of the experiments I want to do. Of course, what we really want to see is a system where we have the multi-probe NSOM or AFM, SPM, seamlessly integrated inside. That would have optimized working distances and all the control things that I struggle with, but I don't think we're actually necessarily going to be that far away. So maybe someday. And so in conclusion, I've shown you we've integrated AFM NSOM into the SEM. We can image transport that way. These are the things we've done it with. You can get a quantitative measure of minority carrier transport, optical coefficients, probably a lot of other things. And I think there's just a lot of potential, both for material monitoring as well as some really nice physics. I want to thank all the students I've worked with. My students at MPS are primarily active duty officers of all branches. Really great work and great people to work with. We've had support from various agencies. I've collaborated with a number of folks. I had a great time as a Fulbright scholar, and I want to thank Professor Lewis and all his colleagues at Nanonics, without whom I would not be able to show you most of these results. So thank you very much.
Any questions? Yes, please.
I come in late, but I really enjoyed your talk. Particularly, I liked your comment about spreading out issues for electrons. But on the other hand, I have to say that both in terms of E-beam imaging or E-beam lithography, we know the convolution can be handled in a proper way. For example, if you consider all the backscattering electrons into practice, you still can generate E-beam features of say 10, 20 nanometers, right? This backscattering does not prevent us to go, to near field in some sense. It is, rather a similar philosophy that I would say, NSOM probe is a convolution imaging, but if we do it properly, we still have a proper access to, surveillance features. So that's,
I agree, but for me, it's not just the E-beam, it's then so I make the carriers, but if the carriers go a long way, that's what's of interest in the transport.
Right, so in that sense, I will say maybe, some, additional features like time resolution could help, but that's a different story.
Yeah.
Okay.
Yeah, if you want to get rid of it, that's true. If what you do, in fact, though, is want to study it,
Sure.
then, I mean, in many of this work, we want the minority carrier mobility, and this is a great way to get it. But you're absolutely right. I'm not suggesting you can't do really high resolution images.
That's Professor Fan from MIT.
Yeah, sorry.
No, thank you. Actually, I asked all my questions during the talk. And I think we should, I guess there's a question there. Yes, ma'am.
I don't know a lot about this, but it seems like the carrier mobility would be directly related to the connectivity of your matrix, right? And so I'm wondering if you looked at different crystal structures or different mixtures, can you correlate that with lattice concepts?
Yeah, so it depends on the problem of interest. So for example, in the example I showed earlier of the anisotropic, the degree of ordering will change that, the crystal orientation will change it, so yeah.
But does it affect your length scale of that propagation by a lot?
Not a huge amount. Right? I mean, it does, but not to the extent that we don't see it at all. It's usually more moderate control. Again, it depends so much on the material system. In the far field systems, I can show you those pictures because we're working with some of the highest quality materials really for semiconductors in the world. I mean, that early diffusion length was 60 microns at room temperature. That's not normal for many of the state-of-the-art systems people want to look at. So that's why we need then some, and with that we can get down to a couple hundred nanometers.
So, but I do have a question, actually. Many years ago, I published a paper which didn't work out very well. Okay? Meaning, it was a good idea, but it had to do with actually the transport distance. Okay, the idea was to generate carriers in a material. Okay, get them through a nano hole. Okay. So the idea would be to actually grow a very, very high quality material. Okay? And then one would be able to shine light on it and use the carriers to actually carry to the end of the tip, for example, and be able to, are the growth conditions they're very complicated for these things.
Well, I mean, now, those are all sort of standard MOCVD kind of growths.
Okay, really thank you very much, Nancy. Now,